Perennial grains, why are we interested in that? People have always eaten perennials, usually in times of famine and other things, but they can be eaten, and there's also oil seeds. We're only about 10,000 years behind wheat in the domestication process and about 500 behind canola. So we've got a long way to go, but we got to start somewhere. As mentioned earlier in some of the talks, they're obligate outcrossers, which means you can't self or mate with a close relative. Genetically, they're actually even more complex than wheat in that it's an auto-allo hexaploid. In Manitoba, when I first started this project, I was told the average yield that they were getting was about 500 kilos per year, and they could do that for about 3 years. This is what happens when close relatives mate in the grass world. If you even get a seed, they tend to be abnormal. Obviously, a plant without pigmentation, with green pigmentation, is not going to grow. It'll maybe put out 2 leaves and die. And because you can't control who they cross with, quite often, even within a line, and those 2 plants standing side by side, one like me, being relatively short, and one like most of my brothers being extremely tall, those are coming from the same parent mother plant. So we have issues with crop height and things along that, even within lines. Manitoba is also known, as is most of Western Canada, for winter. Winter peg got its name for somewhere. And we brought in a lot of materials from the breeding programs in the US. One year, we had a -9.5 frost. I think it was more like a freeze. And we lost over 60% of the materials we brought in. So the materials really realized they weren't in Kansas anymore. This is actually one of the luckier ones. This is what most of them look like. These images on the right were taken in June from a -9 frost in April. So you can see how little these plants recovered. And because of that, when you look at how our materials, the blue materials, line up with the green materials on this chart, we only did one selection cycle, and we got that far away from the other materials. That is why we did some work on trying to figure out how the plant grows. It is a dual induction. Think of winter wheat. Has to go through a winter in order for it to flower. Same with this material. And you can see that the secondary induction, they think, is somewhere between 13 and 14 hours of daylight. I think we killed off all the 13-hour ones with our -9 frost, and we were left with the 14-hour ones, and about half the material that went into developing this model came from Manitoba. So we're a little undecided as to where it belongs. So we sent our materials to be tested against those other programs. And if you look at seed size, most of the Manitoba ones, along with the Minnesota, were at the low end. And these are grown on space plants. And if you start looking at seed yield per plant, it really had nothing to do with seed size. And our yields per plant were anywhere from pretty close to the top down to the bottom. But when you look at it in a plot, the Manitoba ones came to the top, and that's because we did our selection on yield per unit area of the plant. Some plants grow big, some plants grow smaller. And so we took crown diameters and figured out the yield per unit area and made some of our selections based on that. We've also done some crossing of individuals, so we've taken 2 individuals that aren't very closely related. These 2 were different in seed size. One had 10 grams per 1,000 seed, and the other had about 7. And the blue individuals are from the higher plant, the gold ones from the lower, and over half were better than the lower parent. Well over half were better than the lower parent, but we still had some that were down below that. So we need to do a fair bit of work on getting the genes that we want in here to be able to increase that seed size consistently. Because if we had planted these materials out, even just from the blue line in a field, we would have seen quite a variety in seed size. We also did some molecular work. Someone was talking about genotype by sequencing. These are those crosses I showed on the previous page. We were able to map 20 out of the 21 chromosomes. Each of those lines is for a trait. We found traits for seed size. And the other thing you need to realize, we can identify genes, but how heritable are they? Are they high heritability or low? Things like seed head number per plant was in a very low heritability, so we wouldn't be confident of passing along that trait to the progeny. But seed size, we had a relatively good heritability, so if we can identify that gene and get that gene into the individual, then we would be fairly happy and probably fairly successful in bringing along seed size. Ergot is also an issue in this, as this is meant to be a food crop. Again, because it can't self-pollinate, any early flowers, the first ones to open on a plant, are wide open, and any that are the last flowers to open, there's no other pollen coming in, they are susceptible to this, and usually we only see any significant ergot in wet years, and usually on the later developing heads, not the earlier ones. But ergot can be problematic. You can see how big that ergot body is in relationship to the spikelet and floret size. As in most perennial grasses, it has a juvenile year. The one in front was seeded in the spring of the year this image was taken. There are a few seed heads there. You can see a few on there, but the one in back was seeded the previous year. It is the same population that has been seeded in both studies, and you can see that it does need that first year. And because of that first year, we in Manitoba, because we get approximately 500 mils or 20 inches of rain a year, can underseed it to about a 2/3 crop of wheat and not greatly impact yield the following year. So you have some payoff in the year of establishment. This is the 5th year of this study, and I'm taking this one actually into its 8th year this year. You can see that it's growing relatively well. It's fairly aggressive. We've had relatively few weed issues in these studies, even though we're on a farm that has both red root pigweed and green foxtail herbicide-tolerant materials. They come up like you wouldn't believe in the year of establishment. We usually, if we've underseeded to wheat, can use wheat herbicides on most of that. And if we don't, we just mow it off when the weeds are close to flowering. And we've had relatively few weeds being able to survive. Of the perennial grasses, only smooth brome grass has really caused us any issues over time. Now I'm quickly going to go through an experiment we ran across Manitoba and Saskatchewan. We had 2 seeding years at 4 different sites. Our plot sizes were anywhere from 60 square meters to a hectare. So we're not talking about research plots that are 6 meters long by 2 meters wide. These are relatively larger, mainly because we're dealing with populations. We had 3 treatments, 50 kilos of N as a urea immediately after harvest, and just before a rain, we watched the weather. So wherever Regas is, we did it the right way. And that's all the fertility we ever put on any of these plots. We had a 0 or a control, no fertility added, and an intermediate wheatgrass alsike clover mix, mainly because we had tried alfalfa previously, and alfalfa just kicked the snot out of our intermediate wheatgrass. We weren't putting fertilizer on those plots, and the alfalfa just took them over in 2 years. Sweet clover did the same, and the intermediate killed the white clover. So we tried alsike clover, and then we got the 2 driest years on record. Go figure it. We did biomass and 50 random heads to get 1,000 seed weights and things like that. Those are the sites, and I know you're much further north and west of us here, but we're trying to get an idea what's happening across Western Canada. And actually, Dr. Bill Billigatu ran the clavette site for us. This is what the 1 hectare plot sizes look like. This is where we turned to seed them, but we had relatively good catch on this area. And because of difference in seed head heights, we had to swath the whole field. We did harvest approximately 300 square meters of plot on this one to get our yields from it, and then we hired the farmer next door to do all this work. And they combined it, and that was very good feeling to see the grain coming up out of the top of the, even though I've been told I shouldn't use a green machine. This is what the farmer had, and that's what we ended up using. It's very satisfying to see that coming out of the machine. And what we did after that, as we renovated the fields, we baled off the straw. This is 2021. We sold that straw to a producer who came and picked it up. We actually made money off this field that year, believe it or not, and we fed the 9-month-old weanlings on there through the fall. The control plot is the alfalfa that's in the upper left-hand corner there. It's an alfalfa tall fescue mix, one of our better fall grazing treatments that we have for the province. And the gains on the grass we rotationally grazed it, were 600 grams to 800 grams a day on those animals, and on the alfalfa they were in the 500 to 600. So it's as good a stockpile as alfalfa. We started it in October grazing. We were going to graze it again the next spring because we still had other paddocks that hadn't been grazed. And the alfalfa was approximately a foot tall. Beautiful leaves, looked really nice and healthy. And then the Red River decided it didn't like our plot areas and flooded the hell out of us. Anything that was underwater for more than 2.5 weeks died. On the intermediate, that wasn't covered for more than 2.5 weeks. Actually came back quite nice. Unfortunately, it was 2 acres out of a 48-acre field. We did have grazing that spring. Unfortunately, it was carp. We have images and videos of the carp in the field. So if you have a market for carp, we can actually do this as well. That was protein, I guess. So here's some of the results. The first year yields for biomass. The first year harvest was 2020, which was the beginning of our drought in Manitoba, except for the Glen Lee site was actually harvested first time in 2021, which is why we don't have a second year harvest. You saw the previous slide. Second year was 2021, where it was dry. And I mean, we had cracks in our soil that we put a 2-meter stick down, and it was still not at the bottom of the crack. We are on a Montmorillonite heavy clay in the Red River Valley. In year 3, our harvest jumped back up, and fertility after harvest, we don't fertilize till after the first seed crop is produced. We make sure that the area has adequate fertility to grow a forage crop on in the year of establishment. So after that, we only added 50 kilos per hectare per year. The inflorescence density, we're aiming for at least 400. 600 is probably 682 that we saw at Carmen in the second year is probably a little higher than we want, but we'll take it. It's better than a lot less than 400. And in the third year, we were still hitting our target of 400. Grain yield, again, the first year yields were under droughty conditions, and the Brandon yield got hit by cutworms, and I'll show you that on the next field. That was 2020. It was grown on AAFC Brandon. which meant we had to abide by, and that was the first year of COVID, we had to abide by any restrictions they put on us, and that was you were allowed on the site once every 2 weeks. And so we went there 1 week, everything looked really nice. Went back 2 weeks later, and it looked like it had droughted out over about 2/3 of the field. So the yield was definitely impacted by that. The next year, they had close to normal precipitation, which was just over 400 mils. Unfortunately, over half of that fell within 1 week period. Last week of July, first week of August, really didn't do anything for the grain yield at all. So if it's going to be dry, this isn't going to work either. That's the moral. But in the third year, our yields were back up. And on the bottom there, especially where we had fertility applied, we average about 200 kilos per hectare per year. And we've taken it now through 5 years. I haven't shown that data. And we're going to take it through a full 8 years to see if this trend continues. Here's what cutworm damage looks like. And even though the one on the right, it looks green as grass, well, it is grass, even with about a 5 to 10% connection, it still remained green and tried to grow. So I'd wish the worms had just eaten the whole thing off, the cutworms had eaten it off, and then I could have done that. But very little seed came out of these heads, and when we looked at the seed weights in those first few years, they were rather low at the Brandon site. Time of seeding, and we have, like I said earlier, we usually underseed to wheat. It has had no impact on grain yield the next year, even in droughty conditions. And our fall seedings, we've only ever had one that gave us a good enough stand to harvest, and that was in 2019 where we had good moisture. In 2020, again, we did it in the drought. Nothing established, so we ended up with no fall seeding. The big thing here is if you look at the number of heads per square meter, they're lower than our ideal 400. But if you look at the yields between the spring and fall, and then look at the seed head number, there's something else going on there in that the seed heads aren't anywhere near as large or producing as many florets as the spring seeded ones. Here's a visual to show you what the fall and the spring, this is taken the year after. This is the fall seeding on the right, the spring seeding on the left. Fall seeding, hopefully I can see a few more weeds in between the rows than I can in the spring seeding. The spring seeding just jumps out of the ground, and by June 1st is usually, well, as high as my knees. I don't know if that's tall or not, depending on how you want to look at it. But the fall seeding just isn't as vigorous coming into the spring as the fall seeded one. As the spring seeded one. Harvest index, this is something we really have to work on. We have to get it higher. But the Brandon site, the drought and the cutworms really knocked that back. The best we've seen is this here at a little over or close to 11%. And the fertilized treatments a little better than the non-fertilized. What I meant to say back on the yield slide with the fertilized, that's the one we're going with at Manitoba, although I'll show you something in a minute that has me questioning that. What triggers successful inflorescence production? Getting those numbers high enough. If we look at all the environmental factors, fall growing degree days after harvest is highly correlated with the number of seed heads you get the next year. So that's seed head number. So that's one thing. So the later you harvest, the less likely you are to get a seed crop or a good seed crop the next year, which is probably the same for most things. Florets on an inflorescence, regardless of how many inflorescences you have in the area, that's controlled by May and June precipitation. So seed head number is controlled in the fall, floret number in the spring. Two different environmental parameters are going to impact your seed yield. So I think Dr. Brillman talked about drought during flowering. This is our anthesis in 2023. 2023 was a miserable year. I don't know what it was like up here. We had something like a third of the precipitation we usually get in May and June. Really, really dry and hot. And so you can see that even though these are spikes, we have some elongation of the rachilla, and it looks like they may be quasi-panicle. Well, all of those florets fell off. And if we look at what we took in at harvest that year, this is the size of the seed heads. Not very large. A lot of them had shattered, or had broken off long before harvest. If we look at the ones flowering, and unfortunately those aren't at the same scale. The one on the left is enlarged a little more than the one on the right. If we look at that same tray with flower at anthesis the next year, you can see that the heads were quite a bit larger, and we had almost 4 times as many florets per unit area, per square meter in 2024 as we had in 2023. So your growth conditions are really going to impact what you get. We are also starting to use some digital analysis and drone work on our plots. We're also looking at fertility. We've always used fall fertility on these, mainly because of experience with growing a number of these crops over the years. But this year, there was a paper out of Europe showing a split fertility actually does better. So we decided to try that, and we've got 4 rates, 25, 50, 100, and 200 kilos N per hectare, and 3 timings. The first one being fall, which is the F on beside the numbers, a split, which is the S on there, and we're going to put a spring fertility on there. We think we're going to get more, our experience shows us we're going to get a better hay crop and less seed off of a spring, purely spring fertility, but we're hoping that the split will give us some sort of boost in grain production. We've tried some, we used a thermal rating for this, and we've been able to fit an equation line to it quite well. I'm working with a couple of more drone people trying to figure out how this relates to seed production. And we'll follow this through harvest next year, and we will probably, this year, we'll repeat on the same plots, the same trial again this fall, and see if we have something that's better, does it continue to work over time? Because a one of may not be indicative of its long-term efficacy. What can you use it for? Well, you can make bread, and you can make beer, so most people are happy one way or the other. And we're also trying to make whiskey. Apparently the owner of this distillery is very keen on it because of the perennial grain nature. Now this is all from the intermediate wheatgrass. The big thing is you need 900 pounds to make a mash that you'd only use 800 pounds for other grains, mainly because of the seed size, so the starch to protein levels. They say it's one of the nicest moonshines they've made. Those of you who like moonshine maybe can advise me on that as well. I don't know, but it's been in a cask now for 2 years. They're hoping to, I was told, decant it for Earth Day this year, maybe have some publicity around a sustainable whiskey. We'll see how it goes. These are all the people that have been working on this with me over the last only 6 or 7 years, including Bill, should be noted there. Yeah, he is. We've had grad students working on the projects, a lot of technical support, and a lot of collaborators. And I'm sorry I don't have the U of S logo up there, Bill. I couldn't find it quickly today. And with that, I'll let you have any questions that you would like. I'll go back to the mic, I'm sorry. Where we've had problems with it, it's germinated right after shattering. So we haven't had any dormancy in that respect. We usually harvest it and plant it the next year without any issues. I don't plant in the fall. Your second question?
How many species of wheatgrasses are there?
Way too many. It depends. Like we named a lot of things wheatgrasses, maybe 100 years ago. Now they're separating them all out into all these different genuses. When I went to school, which I took my forages class back in the early 1980s, everything was an agripyron, you know, a wheatgrass. Right now, the only one that we have left in agripyron is crested. All the others have moved somewhere, sometimes 6 and 7 times. If I look up under scientific names for intermediate, I've got 19 different scientific names I have to look at. The molecular taxonomists are having a field day. Tall fescues moved 3 times since 1990s. It went to Lolium and then Shedanoris, and now it's gone from Shedanoris phoenix to Shedanoris arundinaceus. And meadow fescues moved with it, and creeping red's still over in the festucas. Go by the common name, it's easier. By the way, intermediate wheatgrass is marketed internationally as an elytrigia. I found out when I tried to send that to Sweden 3 years ago, and they said, well, there's no such species. What the hell are you doing? They made me change the name on it.
Well, and that's the name.
Who changes the scientific name? Taxonomists. They go in and they say, oh, well, who did it start with? Dewey and Barkworth, I guess. They went in and they looked at all the genomic relationships and said, oh, those are no longer agripyrons, get them out of there. And now they've got the geneticists. I'll let Leah, she's a better explainer of it than I am, but why I know why, but it makes things much more confusing for anyone.
Whether you're a splitter or a
So like most religions, right? Splitter or lumper, right? Yeah.
it's a mess.
Yeah. And I was at a meeting 10 years ago where the taxonomist was all excited that they'd moved tomato into the same genus as potato and said, now the breeders can make the crosses. Well, the breeders have been doing that for decades and decades. They didn't need a taxonomist to tell them it was okay. They've been doing it all along. So, you know, I think people realize the relationships or non-relationships as well. Anyhow, there are some different, even within intermediate, there's some quite, Bill tells me mine looks nothing like any of his intermediates. So it's a rather broad leaf and a few other things. So what you select for sometime is what you end up with.
Thank you very much, Doug. This is for you.
Thank you.
That was very well done. Thanks.